Once the radioisotope has been produced, it is transported to the radiochemistry laboratory for the production of radiopharmaceuticals. The two sites could be in the same location, which is typically the case for short-lived radioisotopes produced using generators or cyclotrons. The long-lived radioisotopes can be transported for radiochemistry to distant locations. The exact specifications of the calibration date, activity concentration, specific activity, solvent, pH, storage conditions, radiochemical purity and radionuclide purity are provided in the calibration certificate for the radiochemist. Other information such as decay mode and the emitted energies may also be provided. Typically, the radioisotopes obtained for radiochemistry are in solution, allowing the required activity to be transferred via a pipette to the reaction mixer. In order to produce the radiopharmaceutical, the radioisotope is bound to a pharmacologically active compound or biological component using different synthetic mechanisms. This step is called radiosynthesis, and it is performed in the radiochemistry laboratory. A general concern regarding radiosynthesis is the time required for completion of the reaction. This is because time available for the radiosynthesis is restricted by the specific half-life of the radioisotope. Moreover, the transportation from the production site to the radiochemistry lab has to be taken into account. The time needed for these two steps cannot be longer than 3 half-lives of the radioisotope itself. Hence, there are logistical limitations on how far the radioisotope production site and the radiochemistry lab can be. The synthetic technique used for radiolabeling varies depending on the physical or chemical state of the radioisotope. Radioisotopes from cyclotron could be obtained in solid, liquid or gas form. Accordingly, the subsequent reactions could be in liquid or gaseous phases, each of which have their pros and cons. For example, in the production of carbon-11 labeled methyl iodide, the two-step liquid phase reaction is more reliable, but the gas phase reaction produces higher molar activity. The radioisotope is chemically bonded to a pharmacologically active agent to form the radiotracer. The type of bond, covalent or ionic, depends on the electronegativity difference between the radioisotope and the element it forms the bond with. Radioactive metals or radiometals are generally bound to organic ligands using ionic bonds. The electrophilic metal ions bind to electron-rich donors such as carboxylic acids or amines. The number of donor groups in a single ligand which bind to a metal ion is referred to as denticity, and it can be mono, bi or polydentate. The formation of multiple bonds between a metal ion and donors within the same ligand gives additional stability to the metal complex. The process is referred to as chelation, and the metal complex is called a chelate. An example of metal chelates is the gallium-68 labeled PSMA-11 molecule, which is used for prostate cancer imaging. The PSMA-11 molecule has two specific regions, one for binding to prostate-specific membrane antigen, which is overexpressed in prostate cancer, and the other to bind with gallium-68. The molecule contains 3 donor groups that confer good stability to the metal complex. The rate of formation of the metal chelate, which is kinetics of the reaction, is also an important parameter for radiometal chemistry, especially for short-lived isotopes such as gallium-68 that has a half-life of only 68 minutes. In the case of PSMA-11, the reaction takes about 2 minutes, which is advantageous while working with a radioisotope with such short half-life. Another example of metal chelates is the antibody tacatuzumab conjugated with the macrocycle DOTA. DOTA is a very common chelator for therapeutic radiometals such as yttrium-90 and lutetium-177. Yttrium-90 and lutetium-177 labeled tacatuzumab can be used for cancer treatment. The antibody targets the cancer cells, and the therapeutic radiometal causes damage to the tissue and eventually cell death. DOTA is a good chelator also for binding some diagnostic radiometals such as indium-111 and gallium-68. Other types of chelators used for radiochemistry are NOTA, NOTP, BCERA, for example. A common feature of these chelators are the carboxylic functional groups which bind to the radiometals. Each radiometal prefers a certain number of carboxylic acid functionalities, and hence a variety of chelators are required for different radiometals. The attachment of macrocycles to the pharmacologically active molecule and the final chelation chemistry are fairly standardized. One of the disadvantages of the chelate functionalization is that the large chelating macromolecule sterically hinders the biological activity of the pharmaceutical it is attached to. The problem is greater for smaller pharmaceuticals such as small peptides, where the ratio between the chelator and pharmaceutical size is large. On the other hand, attaching a chelator to an antibody does not significantly hinder its binding efficacy. Radioisotopes which form covalent bonds can be used for substitution reactions, which is the removal of one functional group in the precursor molecule with another containing the radioisotope. To give some examples. The most common PET imaging tracer, fluorine-18 labeled D-glucose, called FDG, is obtained by substituting the electronegative triflate functional group with a nucleophilic fluorine-18 anion. This reaction is an aliphatic nucleophilic substitution. Whereas the electrophilic aliphatic substitution can be exploited to synthesize iodine-124 labeled dopamine transporter. This is achieved by substituting the electropositive tributyltin functional group with an iodine-124 cation. Substitution can also be performed by prosthetic groups that have been pre-functionalized with the radioisotope. A good example of such reaction is the synthesis of a Bolton-Hunter reagent. The reagent is synthesized in oxidative conditions via electrophilic aromatic substitution with radioiodine. The reagent itself can be conjugated to biomolecules in mild reaction conditions. This strategy isolates the harsh oxidative reaction conditions from sensitive biologics like a peptide or an antibody. The advantage of performing a substitution reaction is that the molecular structure of the final radiotracer is very similar to the parent molecule, and hence it is able to mimic its biological activity. The substitute radioisotope can be chosen such that it mimics the moiety that it chemically replaces. For example, a fluorine-18 ion replacing a hydroxyl ion have similar hydrogen bonding capability, and those are called bioisosters. Many pharmaceuticals have fluorine in their structure, which can be isotopically replaced with fluorine-18 for imaging. This is the case of afatinib, which is used for treatment of non-small cell lung cancer and other solid tumors, and can be radiolabeled with fluorine-18 for PET imaging. The development of a new radiopharmaceutical follows a specific protocol. It starts with the identification of a lead structure among molecules with a biological activity towards a chosen target. The selected molecules are used to synthesize radiochemical compounds. Then their pharmacological activity is evaluated using in vitro cell studies. Successful radiolabeled compounds are used for preclinical experiments where further validation of in vivo target specificity, clearance pathways, and metabolic stability is obtained. Based on animal experiments, the candidates with the best imaging or therapy characteristics are chosen for clinical trials in humans. Before clinical use on patients, the selected candidates must be approved by the Drug Administration as a radiopharmaceutical. We have now seen how a radiopharmaceutical is prepared. Together with medicinal chemists, the radiochemist selects the candidates among a group of compounds with desired pharmacological activity and feasible pharmacokinetics. Using this understanding, the modification sites on the molecule are identified to incorporate the radioisotope using appropriate chemical strategies to synthesize the radiotracer. The most promising variants of the radiotracers are passed through several pharmacokinetic, preclinical and clinical experimental phases to be finally approved as a radiopharmaceutical for clinical use.